All right, so in today's video notes, we are going to dive into the process of protein synthesis. It's quite a detailed process, so we're going to try to take our time with it in the next several days to kind of figure it out. But before we begin, let's quickly review where we have learned so far, what we have learned so far. So remember, DNA looks like this. It's called deoxyribonucleic acid. It is a double helical in shape, so it twists like a staircase. And so what we think of as a staircase ladder is that the sides of the ladder are made of a phosphate and sugar alternating each other. So phosphate, sugar, phosphate, sugar, phosphate, sugar. And then the steps of the staircase are our nitrogen bases. Remember, adenine always matches with thymine, and guanine always matches with cytosine. And so let's remember what DNA is made of. DNA is made of essentially just repeating nucleotides. So again, like I said, the sugar, the sugar is down here, and the phosphate, and then the nitrogen base. And then it's doubled, meaning that you have another nitrogen base, a sugar, and a phosphate. And they're just repeated. So it's just repeated over and over and over. And then these bases are bonded with a hydrogen bond. And then it starts beginning to twist into that staircase ladder. So again, that is kind of the structure of DNA. And we kind of talked about DNA replication, which is making an exact replica copy of DNA so that we can make new cells in mitosis. And so this is a picture kind of demonstrating DNA replication again. Remember, there's the enzyme helicase that kind of attaches and unzips the DNA. So look, it's open. And then we have a completely different enzyme called DNA polymerase. Remember, it ends with ase, so we know it's an enzyme, and enzymes are proteins. So this protein enzyme is attaching and like zipping down and adding nitrogen bases to the other pair. So then when this is complete, you'll have two identical sets of DNA. So let's just review the A, T, C's, and G's again, that we know which nitrogen base matches which nitrogen base. So just pause the video real quickly and tell me, or tell yourself, what letters should be under these squares. So it should be T, C, G, A, C, and T. Remember, adenine matches thymine, guanine matches cytosine, and then the same thing. Cytosine matches guanine, thymine matches adenine. All right, so we also reviewed this new molecule called ribonucleic acid. It's called RNA, and we talked about the similarities and differences of these two. So first of all, they both have phosphates, but their sugars are different. DNA has a deoxyribose sugar, and RNA has a ribose sugar. Also, their nitrogen bases, they both have cytosine, they both have guanine, and they both have adenine, but they both don't have thymine. DNA has thymine, but RNA has something called uracil instead. So that's how and also you can see that RNA is singular. It is not a double helix. It's a single helix. So those are some similarities and differences between RNA and DNA. All right, so what I want you to make sure you have with you is your Google Doc note guide. So please make sure you go print that or save a copy, make a copy of it so you can type right on it as you watch the rest of the video notes. So we're going to talk about how you take DNA and you go and make a protein. So this is kind of the flow. You start with your DNA, then you go through a process called transcription to make something called mRNA, which we learned in video notes last time. And then you take your mRNA and translate it through the process called translation to make your final protein. So remember, traits are determined by our proteins, okay? And those proteins are built based off of the instructions decoded by DNA. So we need to decode DNA. We can't take DNA out of the nucleus. It is too precious. It is too important for the cell. So think of if you had this important document, or let's say you had some scandalous information about the President of the United States. Are you going to give your original copies to the newspaper? No. You are going to make a copy of it because it's precious information that you don't want to get destroyed. Same thing with DNA. DNA is precious. and it cannot leave the nucleus because we don't want to destroy it or damage it or get lost or broken or something like that. So DNA has to stay in the nucleus. So we have to decode it through these processes. So we're going to talk about transcription first, going from DNA to transcription to mRNA. So transcription, think of transcribing. We are transcribing an RNA molecule from the original DNA. So it's similar to DNA replication, except for we're not making two strands, we're just making a single strand. And this takes place inside the nucleus because that's where the DNA is. So it's basically kind of writing down a recipe on a note card, copying it from your favorite book, and then giving it to a friend. It's kind of like that. So let's talk about the process of transcription. First, we need another enzyme. Enzymes are crazy important if you haven't noticed in this unit. RNA polymerase is an enzyme that's going to come and bind to the DNA where there's a promoter site, where it's kind of like, here, ding, 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 come and bind to me right here. And it's going to begin to unwind or open up and unzip the DNA. Then the RNA polymerase is going to assemble by zipping down one side of the DNA, not two, just one, and then adding the nitrogen bases, adding those nucleotides. Now remember, it's going to add a uracil instead and replacing of the thymine. Okay, so you can see here that it's just going to zip down, and as it's zipping down, it's reading the DNA and adding matched nitrogen bases to make your mRNA strand. All right, so then the next thing is that once the DNA spot has reached a termination spot on the DNA, then the RNA polymerase is done, and it's going to stop and it's going to leave, and then now you have this mRNA, this messenger RNA, basically the instructions to code to make a protein. So it's basically the recipe. All right, so now that we have our mRNA, now we're going to go through the process of translation to finally make the protein. So the messenger RNA has left the nucleus and now hanging out in the cytoplasm. So when we think of translation, think about translating that message, that mRNA strand that it originally got from DNA. So think about how now that has to be translated. So first step of translation is that we need a ribosome, and the organelle that makes protein. So that ribosome is going to attach to your mRNA strand, and it's going to start to begin to read it. Now it's going to read it or follow it every 3 nitrogen bases. So like an adenine, an adenine, and a thymine. It's going to read 3 letters at a time. And so we call that a codon when you have 3 letters together. So then, each time the ribosome passes a codon, then a tRNA molecule is going to come in. Now these tRNA molecules are just floating around in the cytoplasm. And you notice this is what a tRNA molecule looks like. It has 3 letters. It is the anticodon, essentially matching the codon, and it has the amino acid on top of it. So tRNA molecules only carry 1 amino acid at a time. All right, so in step 3, the amino acids, once they become a part of this process, begin to bond together, and they bond forming a peptide bond between the amino acids. You see they're starting to become a chain of amino acids in this process. So then there'll be a stop codon, which is like 3 letters at the end of the mRNA that basically tells the ribosome it's over, the protein has been made. And so then you'll have a huge chain of amino acids, and that'll fold into itself and become the protein. All right, so let's just remember real quickly about proteins. Remember, they're made of amino acids. And there are actually only 20 different amino acids that exist. And so how do we know which amino acids go with each mRNA sequence? And so we use something called the genetic code. This is what a genetic code looks like. It's a little intense, but I'm going to teach you how to read it. We're going to use the circle one. So first, you want to read your mRNA strand by every 3 bases. So you have these 3 letters, A, adenine, uracil, and guanine. That's called one codon. This is another one, and this is another one. Then you're going to use the genetic code to find the amino acid. So let's say this is our mRNA strand. So first, we need to kind of separate them out by threes. So these three letters are together, these three, these three, these three, right? Okay, now that we have these codons, we're going to find out what amino acid goes with them. So we need the genetic code, we need this. So we go AUG. So A, the first letter is this one. Then the second ring is the second space. And then the third ring of letters is the next place. So let me explain by showing you AUG. What amino acid is AUG? Well, A is in this one. So now we're only going to focus on the amino acids in this box. And then it says U. So now here's my U. So now I'm only going to be zoomed in on just what's in here. And then it's G. So here's G. So my amino acid is methionine. Okay? All right, so let's try CCU. Okay? CCU. Pause the video and see if you can find it. All right, so C, so we're only going to be in this area. And then a second C, so here's my second letter. So now I'm only going to be in this spot. I don't even have to look at the third letter, but it's U. So I'm going to be at proline. Proline is going to be my next amino acid. Okay? All right, so then GGA. See if you can pause and do it. GGA. So guanine. So now I'm only going to be in this box. And then G. And then A. So I'm going to be in glycine. Glycine is going to be my next amino acid. And then CAU. CAU is going to be in histine, a histidine amino acid. And then UGA. UGA is going to be my stop. So I have, this is my amino acid sequence. This would be my amino acids to make the protein that codes for this gene. Now, this is a little bit of a misconception because our mRNA strands are quite significantly longer, and proteins have a lot more amino acids in it, but this is just kind of a general way of learning how to read the genetic code and which amino acids go with which proteins. So what I want you to do now is take a minute and go back and review your notes, and then I want you to go to today's formative assessment questions, and I want you to please try to use your notes for support and help with answering those questions today.